If there's anything else. The next chapter, 14, is going to also be broken up into two days, right? So we'll split it in half. It should be a lot in that chapter. I'm thoroughly at it, but it's about all the tech are useful for. The chapters that follow are going to be, in my opinion, a lot lighter and I'll cover every topic in there. Okay, so last time when we did part 1, I assigned a bunch of practice problems, and we're just going to go over one of them today. Is there a little? I didn't think it was too. I don't think you guys might find this too difficult, but the difficult part is just all the different types of mutations, right? All the different possible outcomes of the mutation, and kind of being able to learn which is which and how to be categorized. So this is problem number 18 that we're going to go over. So it gives you an amino acid sequence. Okay. And then it gives you a bunch of different mutants. So it wants you to do two things. It wants you to indicate the type of the mutation that occurred in the DNA. All right. So look, we have a amino acid sequence. We're definitely going to need to make use of the codon usage chart to get a DNA sequence. Now, does the codon usage chart have DNA on it? What is it? Yeah, it's an mRNA. Got to keep that in mind. So they're also real specific in the question about what type of mutation is occurring. It's just giving you 3 choices here: single base, insertion, and deletion. Now with substitution, remember there were 2 types. What were the 2 types? Transition and reverse. Transition and transition. Maybe you could elaborate on that if you needed to. But they left the second part a little more open-ended. Phenotypic effect, they have the word abbreviation, et cetera, in there. So that would lead me to believe maybe there are other possibilities here, right? Let's take a look. All right, so the first one, we're going from methionine, serine, proline. And if you look, so we've got another serine here, okay? And the rest of the sequence, arg, leucine, glutamate, glycine, is intact, right? So we've just changed a single amino acid. How do you do that here? So you need to refer to the codon usage chart. Oops. All right, so the serine, if we look, here's serine. They all start with U. And the proline here, they all start with C. So, and if you see this commonalities between the second nucleic acid and the third nucleic acid in the mRNA, right? So, looking at that, changing just the first letter would be significant enough to change the amino acid from a serine to a proline. So this would be a type of missense mutation, right? That would describe the phenotype because it's actually resulting in a change in the amino acid, right? And it would be a single nucleotide substitution, right? We're going here from U to C, the mRNA. The question's not asking us to provide the DNA. Indicate the type, yeah, just indicate the type of mutation. So and then it's going to be transitional, right? Yes, transitional. All right, this one's pretty easy. Look at the next one. Okay, here we've got same sequence up there, and look, it's shorter now. So we've got a methionine, a serine, and a proline. So if you have a truncated protein, that is indicative that you have a sort of nonsense mutation, right? You have the introduction of a stop codon. And it has to occur in that arginine. So if you look at the arginine sequences here, now, if you didn't know this already, I don't know if you guys can see this, but arginine has more than 4 codon and anticodon sequences. There are 2 more that are indicated right here, so that might trick you up. But you can go from the CGA here, right, to a UGA here by just changing the first nucleotide. Or you can go from a AGA to a UGA by just changing the first nucleotide. Those are the only 2 possibilities that I see there to include a stop codon. All right, any problems with that? So just something when I was looking at this, you know, there are more possibilities of mutations, things that we went over that could account for this. Can anyone think of one? Done a single base substitution? What else could put a stop codon in there besides a single base pair substitution? Of what?
3 codon?
Yeah, you can get an insertion of more than 3 codons, right? We had a specific name for that. We could get, you know, segment of another gene, chromosome could come in. There's a lot of different possibilities, and that was why they kind of gave you 3 to narrow your choices. But that's just something to keep in mind. The question could have been very open-ended, but it, you know, narrowed down. Single base substitution. I guess you could get insertion too of 3 nucleotides, if that makes sense. But a deletion wouldn't work. I mean, you would have to have a deletion and then an additional insertion. That's some possibility. The more mutations you have, the less likely the scenario is. And a single base pair mutation is the most likely than having like a 3 nucleotides substitution. Okay, so just you got to kind of default that bank of different types of mutations that we learned about. Okay, so here we're going from, so we got a big change in the amino acid sequence after the proline. Right, so what could account for a really large change in the continuing sequence? That would be a frameshift mutation. Right, so now when I worked this out, when I was looking at it, I didn't take it so far as to check all of the nucleotides and kind of figure out, you know, what nucleotide changes would account for the changes of all of the amino acids. I just looked at the first one, right? So we're going from an arginine to an aspartamine, right? So I saw that there. I think most likely the CGA, right? You take that off. And the GA becomes the first part of the next amino acid. All right, remember, if you add or remove 1 or 2 nucleotides, you shift the reading frame of it. Any questions about that? Comments? We're good? So here it looks like when you compare the sequences, we lost that arginine and the leucine. So obviously something got deleted out because the glutamate and the glycine remained. Then we have what's called an in-frame deletion, right? So if we only pulled out 4 or 5 nucleic acids instead of 6, then we would have had a frame shift 2. So we would have had a different sequence here of, and you actually would have only had 1 amino acid. You wouldn't have had 2. Okay, so this was a deletion that was in frame because you pulled out 2 codons. Okay, here's your sequence up there. Now we've got 2 leucines. So that can only be accounted, and we keep the 2 flanking amino acids here. So the only way you could do that is an in-frame addition. So in-frame insertion or in-frame addition. So you're adding 6 nucleotides. No, I'm sorry, you're adding 3 because you're adding 1 leucine. Okay. Okay. Make sure that you work the other problems. And what I can do also, I can stick around. I go through the problems. We'll probably get through this chapter a little today because I took some of the hard stuff out for you guys.
Thank you.
You're welcome. Okay, so we're going to talk about mutation a little more. The stuff that I took out was the stuff on the translocations because I think it's a little tricky. But we're going to talk about like what happens more chemically in the DNA that causes the mutations, things that affect the rate, and right. So, what sort of factors affect the rate of mutation? So the frequency of mutation, right? You have to look at not only is the mutation present, but also is there a possibility or what is the frequency of correction of that mutation? And then also look at if the mutation can actually be detected by some molecule in the cell. There are additional proofreading enzymes besides the ones that we learned about when we learned about DNA replication, that whose job is to go along and correct mutations in the DNA. All right, mutations can be spontaneous, right? So this means that they occur under natural conditions, right? So DNA polymerase, it has very good fidelity. It's a mistake less than 1, a little closer around 1 to 1,000,000 insertions. So every time it's building nucleic acid, like there's only a mistake like 1,000,000 times. But it's still a mistake and it does occur. We would consider that a type of spontaneous mutation, right? And also mutations can be induced. Whether it's an environmental toxin or a toxin that's produced in the cell, and then radiation as well. All these things can induce mutation. Okay, if you've taken Gen Chem, you might have learned about tautomeric shifts occur when right, so if you think about in its base region, the nucleotide, if you start shifting the protons, you're going to filament nucleotide, and that can permit comes in the underwent some shift, it's permitting binding, spit it out, pull in the correct one. Okay, so tautomeric shifts can like mutation. Right. Something else we're going to look at is called strand slippage. So what formation of this tiny little nucleotide in size comes into place. Okay. So if you remember from Gen Chem, these shifts occur with very highly polar molecules because they're capable of hydrogen bonding. And they are in a state equilibrium between their original conformation and their shifted conformation. Proton moves, right? It's a state that manifests itself moving back and forth. But they can stay their hydrogen bonding, it won't keep shifting. All right, so the most common form is when these T's can get a shift in hydrogen bonds to basically form a bond like a C, right? So if you had an A in the other position, now you can incorporate a G into the position. So that's the most common tautomeric shift right there. So T to C. All right, the other one is when you get these small little loops that occur. All right, so here you've got an original double-stranded DNA, right? It separates for DNA replication. And if the binding is not perfect, like the hydrogen binding is not perfect, you'll get this area where the nucleotide loops out a little bit, right? All right, then it separates to go through another round of replication, and now you've end up with a mutated form. All right, this is called strand slippage. Oh no, this is strand slippage. What is this? Wobble basing. I think I meant to take this out, guys. I'm sorry. I'm making a mistake here. No, this is the sorry. This is the tautomeric shift there. So you've got a T that's come in, TTCG, and yeah, so a G has come in this position right here. So that's what's shifted. Sorry. Okay, so the strand slippage is a little different. So when you get the synthesized DNA you have a newly synthesized strand, you get a nucleotide that loops out here. Right? So this will ultimately add a nucleotide to the newly synthesized strand. Right? The alternate strand, though, that's where it's not being looped, is going to end up losing one. If I'm getting this right, that gets confusing. So up there, look in the template strand, you've got 1, 2, 3, 4, 5 T's. You've got 4 A's. And here you've got, right, so you have that series of the same nucleic acid that is in tandem, like right next to each other. So when they come to reanneal, like after replication, you can get this looping, right? So when it comes back together, it can loop and it can leave an area that's not fulfilled. So when it does that, then polymerase will come in, it will finish the sequence, right? And you've added a nucleotide into this. That's what the slippage is talking about, right? So one of the strands, it's looping out and it's slipping. So that shows you what's happening on the newly synthesized strand. And then on the template strand, so look, we've got 5 T's here, right? The template strand is slipping. So one nucleotide is bulging out. And then when DNA polymerase comes and does it, this side is going to be lacking a nucleotide. Okay. So strand slippage affects both the newly synthesized and the template DNA. Right? The newly synthesized is going to get one added, and the template is going to. Right? But I don't know of any cases where this affects more than one, which only affects one nucleotide that's bulging out. Okay, so there are some cases that cause as well. We're going to talk about depurination. So this is specifically the loss of the purine group from the nucleotide. So this is not an entire nucleotide that's leaving the DNA or getting lost. It's just the nitrogenous, right, the part that makes the purine. So the sugar and the phosphate remain, and you actually get a site called the apurinic site, which has the sugar phosphate backbone. It's just lacking purine two-ring structure that's in the middle of the DNA. Right? You're going to see that results in like 2 new nucleotides. The way that works. So that's depurination. Deamine group is lost. So this can either occur spontaneously, occurring, or it can be induced sort of chemical or ionizing radiation. So this is a specific type of deamination is removing the amine group from Cs and turning them into uracils. Okay. There's also a version we'll see on the figure where you have a methylated C, and that can turn into a T. Okay, so we've got our DNA over there on the left. You can see the purine is the second nucleic acid. We know it's purine because it's got 2 rings, and it's a G, right? A's and G's are the purines. And it undergoes some sort of process where the rings pop off. So that could be like ionizing radiation. So it breaks the covalent bonds there, and those 2 rings pop off. So now we have what's called an apurinic site. Right? So you still have that sugar phosphate backbone here, but you're just lacking the ring structure. Okay, so those will separate during DNA replication. Right? This thing is still together because you have the phosphodiester bond there. And then during replication, you get incorporation of a new nucleotide right here. And with this type of apurinic site, it always pulls in an A. Okay, so you have to kind of remember that. With apurinic sites, they uniquely pull in always an A. You get the addition of an A there. And then when that separates again, now you'll get a T that forms on the opposing strand. Okay, so that AT combination is unique for apurinic sites, and as a result of that apurinic mutation. How you guys feeling about this? Good? Okay. Fun stuff. All right, so understand that these are like, we learn generally in the first part of the chapter, just generally about mutations, right? Different types. Now we're learning some very specific examples, and yeah, you have to memorize, the information about that specific example. So depurinate most commonly applies to the G's, right? 2 rings, and an A is going to get made in the opposing strand. Result of DNA polymerase reading that empty site. It's going to insert an A on the other strand. That's apurinic. So deamination, also a very specific type of mutation. It changes when you pull off that amine group of a C, and the molecule now looks like a U. Right? So it turns C's into U's. There's another version of C, though, that exists in the cell, the methylated C. And so methylated C, when it pulls off the amine group, it looks like a T. So it just depends if it's methylated DNA or not. But deamination specifically affects these C residues to turn them into U or T, depending on their methylation status. Okay, chemically induced mutations. So these are mutagens that can have different effects. Right? One effect they can have is they can be a base analog, which means that they're a molecule that looks just like the nucleotide. Now, this is usually, there's not too many base analogs in nature, right? These are more man-made structures, and They're just not naturally produced. They have to be synthesized in a lab. 5-bromouracil is a very, when you're trying to induce mutation bacteria, you can introduce 5-bromouracil and it will increase mutation rate in that bacteria. 5-bromouracil is an analog of T. Okay. So not only is it an analog of T, but it can hydrogen bond to G. Okay. So this isn't like a typical U molecule. It's called 5-bromouracil. You're not going to get like natural bromination in the forest or whatever, or in our cells. All right, so this nucleotide will mispair because when it gets incorporated in the DNA and DNA polymerase reads it, it will stick a G on the other side. Okay, that's the actual mispairing. So eventually that, gets lost, but the mutation remains is in that initial piece of DNA. Okay, let's look at this here. So we got our sequence over there on the left, CA was a GAC and CTG. All right, so you get strand separation, and now we've introduced 5-bromouracil. So DNA polymerase is pulling nucleotides, and now it pulls in this 5-bromouracil by mistake instead of pulling in T as it should. So now we've incorporated this error, right? It thinks it's a T, but it's not. So that goes through another round of replication. It separates, right? And when it separates, you can see one side of it replicates normally, but the other side, even though it thought it was a T, it's going to hydrogen bond appropriately to a G, okay? Again, these that you're going to have to memorize to be able to it's on the exam. Okay, so that separates, and now you have an ongoing mutation in both. Right, 5-bromouracil could reincorporate again. Right, obviously it's going to get depleted at some point, but that's why this is a very powerful mutagen, because it's an analog for these Ts, and as long as you give it in like a really high concentration, it's going to incorporate very often during replication. Okay, so remember, it takes the place of T, but it brings in a G on the opposing strand. Okay, alkylating agents. So these are agents that will actually add chemical groups, which are alkyl groups, to the nucleotide bases. All right, so the ones that are mentioned is this EMS, ethyl methyl sulfate. It's a strong alkylating agent, and it will add a methyl group onto a nucleotide. Other chemical inducers include chemicals that deaminate. Right, we just saw examples of deamination. Hydroxyalamine will add hydroxyl groups to it. And then also intercalating agents. Intercalating agents actually like insert and fit in the grooves of the DNA. And it's almost like adding additional nucleotides, right? In there between the base pairs of the DNA. Methidium bromide is one that's really commonly used in labs to visualize DNA because it fluoresces orange. Dioxin is a chemical that results from combustion. It's an environmental mutagen, very powerful mutagen. Acridine orange, that's a chemical used in chemical warfare. And proflavin, I don't remember. Okay, so all these are just different examples of chemical mutagens. They're not the base analogs. They're actually changing the molecule. Alkylation here, it's adding an alkyl group over there. Nitrous acid is deaminating, right? So it's taken off that amine group right there. And hydroxylating is adding a hydroxyl group here. Okay, intercalating agents, these insert into the grooves, right? So here you've got the regular base pairs in the double helix, and the intercalating molecule is just kind of glued in there in between. That's what intercalating. So, you haven't chemically changed anything, but when DNA polymerase comes along and starts reading the molecule, it messes it up. It changes the shape of the whole DNA structure. It might stall or misread when DNA polymerase is on there. Okay, base analogs are mutagenic because of which characteristic? They produce changes in DNA polymerase that cause it to malfunction. They distort the structure. They're similar in structure to normal bases, but they're chemically modified. See? So if they're similar in structure, I would say those are analogs. Do that. Analogs are the ones that are similar in structure. So they'll actually get incorporated like as an A or a T, like 5-bromouracil is an analog. What do you think?
That's the answer.
C is highlighted?
Yes.
It should be B. It is. Oh, no, that's wrong. Okay, yeah, it should be B. They distort the structure of DNA. So they're right there, distort three-dimensional structure.
Analog is the result of mutation.
No, so analog, these are the ones that have structures that are similar to the nucleus, right? So 5-bromouracil is an analog. It looks like T.
That was asking.
Base analogs.
What? Because base analogs are mutagenic because of which characteristic? Yes, you're right, guys. I'm sorry. I'm like half asleep up here. Yes, it is asking about analogs. All right, and C is the right answer for that. If it was asking about intercalating agents, the answer would be B. I was thinking it through. It's up here daydreaming. Okay, a little bit about radiation. Radiation greatly increases mutation rates in all organisms, right? You know that. So there's basically two types of mutations that are types of radiation that we're going to look at. UV radiation, it causes a very specific type of mutation called a pyrimidine dimer. And then ionizing radiation, which is more damaging, like pulls off electrons, breaks apart the molecule, and it causes more specifically what are known as double strand breaks, right? So you're actually breaking the sugar phosphate backbone of both sides of the DNA molecule. All right, so pyrimidine dimers most commonly occur with the T residues, and it's covalently links them together from that ultraviolet radiation. So when DNA polymerase sees it, it actually thinks it's like one nucleotide. So it'll usually stick one A on the opposing strand. And you end up getting possibly a frameshift mutation from that. So there's some interesting biology with these UV pyrimidine dimers. So eukaryotes have evolved to correct these dimers, big enzyme that actually requires UV light. You have UV light here that's causing the mutation, and then the proofreading enzyme requires UV light to actually make the correction. That's not mentioned in your book, but something that you can take home with you there. Okay, so what sort of tests can you do to kind of assess for mutations? So the most common one is the Ames test. And this kind of was developed in the 70s. It was a result of a lot of funding for tobacco research. And it's a very simple sort of assessment that you can do to quickly identify if a chemical is considered a carcinogen, right? This is a chemical that causes cancer, and 90% of all carcinogens cause cancer through mutation using the actual DNA sequence. So it's a test that you do on bacteria. Use these oxotrophic histidine lacking bacteria. So these bacteria lack the ability to make that essential amino acid histidine. You take whatever chemical you're testing. So the liver enzymes are going to metabolize the chemical and then produce what actually is present in the body that could be the carcinogen. And then you plate it on media that lacks histidine. And basically, if the mutation that was in the bacteria was providing the histidine, if it has a forward mutation to restore the type, then the cells will grow. Okay, so you take your his minus bacteria. That means it has, remember, histidine is amino acids, and the cells are going to make it. So in order to do that, they have to have, and this strain of bacteria mutation in the produce histidine. So only in the presence of histidine the cells grow. All right, so you take whatever chemical that you're testing and you mix it up with some liver enzymes. Chemical, that's that one over here. So you mix it up with some liver enzymes to kind of check the metabolites of that chemical. And you combine it with your His negative bacteria and you plate it out. And if the His minus has been restored to His plus, then the cells will grow on the media, and you can assume that that chemical is a mutagen. Did that bacteria. Okay, so 13.3 goes over transposable elements, right? So these we talked about a few chapters back. This is when large pieces of DNA move about the genome, right? They'll move from one chromosome to another, although move within the same chromosome. Right? The pieces that are moving are called transposons. The movement event is a transposition. Okay? So I took out a lot of things, what sort of kind of mutations that are common with these. And really the only one we're going to talk about is what's called a flanking direct repeat. Okay, so here's our transposable element. So it's going to get, it came from some other area of the genome, and it's going to come into our DNA. So the first thing that has to happen in this process is the DNA has to be cut to let that new piece of DNA in. So if it's cut in a staggered orientation, like so you're producing these sticky ends that they're called, right? You have overhangs. And when this thing comes in, there's going to be gaps. And when DNA polymerase goes in to fill it, right, it's going to do so. So you have these flanking repeating sequences, right? Because they're flanking the transposable element. Right? This is something that's consistently seen when transposons move within the genome. Right? They'll move and they'll be flanked by these repeating sequences. All these flanking direct repeats. So why are they repeating? That's kind of the take-home message here. Because it would cut in a staggered orientation, right? This here is a complement to this. And that's how you end up getting the same sequence on both sides. Right? If it was cut blunt, right, then there would be nothing to fill, and you wouldn't get anything repeating. Okay. All right, so what are mechanisms of repair? So there is mismatch repair mechanisms, direct repair of the nucleotide molecule. So base excision repair. This is obviously going to require some cutting. And nucleotide excision repair also is going to require some cutting as well. Okay, so what do these enzymes do? So they basically cut out a portion of the newly synthesized, the fresh DNA following replication, and they just replace it with new nucleotides. Right? So the thing with mismatch repair, though, is it's on the newly synthesized end of DNA. It's cutting out a section of just the newly synthesized strand. What's one way that these enzymes can know what's newly synthesized and what's not? Like, how would it determine if something fresh DNA or old DNA in parent strand?
Methylated.
Very good. The original DNA, the parent DNA, could be methylated. Right? The newly synthesized DNA would be. Thought I pulled this figure. I guess I didn't. Okay. All right, let's work through this here. So in DNA replication, a mismatch base was added to the new strand. Okay. You can see it's bulging out there because it's shouldn't have T as a complement. So the opposing strand is methylated. It's got a methyl group on it. That's how the enzyme knows that this is the original and that this is the newly synthesized strand, that GATC up there. GATC. Okay, so this complex will kind of bind over the whole region. It'll detect what is new and what is old by the methylation status. And then the next step is that exonucleases will come in and remove this strip. You see it loops it up and it cuts this whole region out of newly synthesized DNA. And then it puts the correct DNA in. Right, so there's basically 3 steps to this, right? There's identification of the mismatch. Then there's identification of what the parent and the newly synthesized strand DNA is. And then there's the cutting and the replacement. All right, so direct repair, this is correcting the actual structure of the DNA. So if there was like the addition of an alkyl group, it would remove that alkyl group, something like that. Base excision repair. So there are enzymes that will recognize and remove specific modified bases, like glycosylase enzymes. And in this instance, they remove the entire nucleotide and then they replace it. Okay, so direct repair work on the molecule. Base excision repair removes the wrong molecule and puts the correct one. A good example of this would be methyltransferase. So methyltransferase can remove that alkyl group up there, methyl group, and now you have the correct form of guanine. Nucleotide excision. So this is what nucleotide is in the DNA. It's going to pull it out, and it's going to put in the correct one. And so this is going to require some additional enzymes, such as DNA ligase, to seal the gap that's there when the new nucleotide comes in. All right, and the last thing in this chapter just talks about genetic diseases and their mutations in genes cause disease, but also mutations in genes for DNA repair cause disease as well. So keep that in mind. All right, and a lot of breakthroughs are in fact caused by problems with DNA repair enzymes. Okay. All right, so that's everything in chapter 13. And guess I didn't give you more problems to work with, right? Let's take a look. Definitely work all the applications. You guys have a great rest of the week. I'll see you tomorrow. So, why is it a holidays? Today, Monday, see you Wednesday. See you guys Wednesday, Wednesday for lab, right?
Yeah.
Forward to it.